mode now.
No, that's okay. Are you able to see now?
Not yet.
Just send it to Nitika. Then that ways you can also like allow people in, and Nitika you can just tell her to change the slide. It's easier.
Okay. So people are still...
Yeah, again, your screen is here. You can see the screen.
Okay, what I will do is, hello.
Yes, sir, we can hear you.
Okay, so I'll just speak what I want to say because mine is hardly a few minutes, so let me not waste time on this. Okay, so the aim actually has been to bridge the gap between academic programs, reality, and employability for the youth interested in pursuing biotechnology. So for this, I started a series called Scientists Who Live So That Humanity Lives Longer and Healthier. And in this series, we already have started with Dr. Ella Prakada Subbarao. Then we spoke about Frederick Sanger and Louis Pasteur. The next one is on Karl Landsteiner. So this essentially has been created to, have students. We have formed what is called a Biotechnology Education for Students and Teachers Club, where we hope to give at least 2 lectures a month and 1 tutorial for students who want to know more about or understand what the talk was about. We have also started an internship program, and last month we completed a 1-month internship program, whereby the children had 1-hour theory lecture followed by 2 hours of demonstration, data analysis, and interpretation. Okay, our idea is to make the teachers and students form a sort of network so that we can discuss where are the opportunities available. Okay, so last time when we did the internship, we did the revision of concepts and techniques with experts in the field of protein sequencing, proteomics, genomics, zeptomole fluorescence sequencing. Second week, we had clinical application of hematopoietic stem cells and cord blood banking. Third week, we had one week on protein sequencing, LC-MS, and high throughput proteomics. Fourth week, we had a whole thing on metagenomics, COVID genomics, how to find out the various variants, as well as we had one on cytometry. And the last week, we had the whole session on what is food technology and how bacterial infections are prevented in these kind of things. Okay. Now, since we have experts coming, so we conduct these workshops and internships. Where basically we pay honorarium to the faculty who take the classes. So we charge a very nominal amount, and we normally expect for internship about 20 students, so that we are able to get enough speakers who spend time, and as a token of honorarium, we pay the money for that. Okay. And we want to continue this practice all along. Okay. And I want that after today's lecture, block yourself for 23rd August, when one of my dear friends who has discovered a lot of new blood groups will be talking, Dr. Sanmukh Joshi. Already the circular has been sent to all of you. Okay. So thank you so much. And now I hand over to Nitika to manage this. Thank you.
Thank you, sir. Thank you so much. And it was nice to know about what KKBiotech is doing for the students. It's rightly said that, a mentor empowers a student to see a possible future, work towards it, successfully work towards it. So you've been a great mentor to us at School of Biotechnology. And I'm sure by means of KKBiotech, you will touch and ignite many more young minds. So I wish KKBiotech real success, and I hope the students benefit the most from it. With
Thank you
that,
so much. Yeah.
yeah, yes, sir. Okay. Let's move on to the next part of our session today, where we have an outstanding scientist, Professor Aditi Bhargava, with us. Professor Aditi is a full-time professor with Department of ObGyn at University of California, San Francisco. She started her research career as a research fellow at Ames, where she first worked on human papillomavirus. She was also a part of the team that developed the first PCR-based diagnostic kit to detect Mycobacterium at Ames. Then she moved on to University of Pune for her doctoral work, where she studied, the understanding of normal function of proto-oncogenes. Her postdoctoral work basically involved understanding how steroids and mineralocorticoids cause heart diseases and hypertension, and what role do they play in stress-related brain disorders. So she identified SGK1 as the first aldosterone-regulated gene, and the seminal finding was published in PNAS, and it has already been cited more than 1,000 times. She has many more publications in reputed journals to her credit, and she has scientific associations with renowned societies such as the Endocrinology Society, UCSF OSHA Center for Integrative Medicine. Her research focus primarily has been to better understand the stress-induced pathophysiology and the role of sex differences, if any, in the same. So let's all welcome Professor Aditi Bhargava. She'll be talking to us about making sense of SARS-CoV-2 and COVID-19, the noise versus signal. And I request you all to keep typing your questions in the chat box so that we can take them at the end of the session. Welcome, Professor Aditi.
Thank you very much, Nikita, and thank you, Dr. Kannan, for inviting me and giving me this opportunity to present some of the, or rather try to make sense out of what's going on with COVID-19 and SARS-CoV. So I will start, but hopefully I have better success than Dr. Kannan in sharing my screen. So let's see. Are you able to see or no?
Yes, we can see your screen.
And do you see the full slideshow?
Yeah, I can see the slide.
But the slideshow and not the window of the slide.
I see the slide only.
Okay. So as you all know, that COVID-19 has perhaps brought science to a stage where the general public has been consuming it with the kind of veracity that we haven't yet seen. And there's the media, the mainstream media, the public, the social apps, platforms, everybody's been buzzed with COVID-19 coverage. And in a sense, it's created a lot of chaos. I find there's a lot of obfuscation. The news that's coming out is not quite clear oftentimes. There's been a lot of vitriolic kind of information out there, especially in the mandates that have been passed, first starting with mask mandate, where some set of people didn't want to wear masks and they were sort of stigmatized. And then with the vaccine mandates, what's happening, and people who are not keen on taking vaccines are being forced to take vaccines. In a lot of cases, you're hearing where their parents are being forcibly taken away from them, so there's a lot of misinformation, and the mutations that are happening, not just to the scientific information, but to the virus, is also pretty amazing. So that's definitely created fear, uncertainty, and doubt in the mind of people. and our society. And it's unfortunate that the science has gotten this center stage with this kind of pandemic rather than something more positive. So this whole situation really reminds me of this parable about 6 blind men and an elephant. And I'm sure a lot of you have heard about this, where these 6 men, experts in their own field, but blind, and were asked to identify this beast, but with just the information that they were allowed to sort of, in this case, feel. So one person had the trunk, one the tusk, ears, body, leg, and the tail. And the conclusion each of those experts reached was that the trunk was a snake, the tusk was a spear, the ears were like a fan, legs were like a stump of a tree, the body was like a wall, and the tail was like a rope. So taken out of context, none of them really reached the right conclusion. And this is how I feel the COVID-19 SARS-CoV situation is, that a lot of things have been taken out of context. A lot of information has been distorted, and where we are is in this chaos where there's a lot more noise than there is signal. So hopefully a number of you as students will sort of, instead of consuming this from the media or from tweets, or even just looking at abstract, will actually go and read the paper, look at the raw data, and come to your own conclusions rather than just consuming what's been given to you. So critical thinking is extremely important. So let's look at the history of pandemics. And as you can see, this is obviously not the first pandemic that our society has faced, and there have been many more. This is a nice depiction of a visualization of pandemics. And the death toll from these pandemics, we are somewhere in here, and of course, as it says, it's ongoing, so the exact toll is not known. But as you'll see, there are lots of other deaths that could have been prevented, but will also be clubbed into COVID-19 deaths. And I'm actually not clear as to what is labeled as COVID-19 death. But nonetheless, I'm also not quite clear how these death numbers are calculated, but here we are, where the Black Plague or the bubonic plague so far has caused the most number of deaths. But the pandemics, as you can see, have been going on for centuries. And unfortunately, they seem to have become much more common in the last 3 decades, where we've had one every 10 years, starting with SARS, then MERS, and now the COVID-19. So that's sort of a disconcerting news, given that before it took 400 to 500 years for pandemics to show up. So the first is noise versus signal. Is, of course, there's been a lot talked about the origins of SARS, and is it occurring in nature versus a lab leak? And there's a typo right there, but I just wonder if SARS itself is a misnomer, because that stands for severe acute respiratory syndrome. And as we know, not everybody who gets infected with SARS virus actually gets acute respiratory syndrome. So when this pandemic broke out, it was pretty much a foregone conclusion that this was a zoonotic jump and that it did not come from a lab. And very quickly, a group of scientists organized a letter that was published where they claimed that, or gave their reasons as to why this was not a lab leak, which itself is, it's sort of like, why are you talking about lab leak if it's not a lab leak? So here we are. But if we look at the history of coronaviruses, they've been actually around for a long time. And just recently, I think they discovered a 20,000-year-old human remain, which apparently had telltale signs of COVID or a coronavirus infection. But the virus itself was identified by Dr. June Almeida in London while she was working on nasal samples from medical students who had flu-like diseases, but it wasn't actually the flu virus. And she worked with Dr. David Tyrrell in St. Thomas Hospital in London, and she identified this virus particle, which, because of its spikes, they called it coronavirus. And she had previously seen similar virus particles while she was studying hepatitis and bronchitis in chicken. But when she sent her findings for publication, they actually rejected it, saying that, oh, the images were sort of fuzzy and not, and it wasn't clear if this was indeed a novel kind of virus. So here we are, this lady who helped in identifying coronaviruses. And in the 1980s, she also helped take pictures of HIV. Unfortunately, she's no more. She died at the age of 77 in 2007. So the first SARS epidemic was reported in November of 2002. It spread to about 33 countries. It infected 8,000 people and killed about 800 people. The Chinese wet market was blamed for this zoonotic jump. So, and of course, the Chinese horseshoe bat was thought to be the reservoir for this virus. But even though it was called SARS, because like I said, in some people, it caused a severe acute respiratory distress. But the virus for this was not actually isolated or identified, or rather isolated till October of 2013. But having said that, there were publications that sort of warned about an emerging pandemic because of SARS virus. So I guess it's sort of been foretold in some ways. But having said that, that the virus from the bats were sequenced in 2013, there was this publication, or several publications actually, including one in PNAS shown here, which claimed to have reverse engineered the whole virus. They put together bits and pieces of cDNA for the SARS virus, and they assembled the whole genome, and they showed that this virus was actually infectious. So there's definitely a gain of function kind of work going on, which has been, again, sort of denied in the media, but we know that there is a gain of function studies that go on all the time. So again, this identification or isolation of SARS coronavirus that used ACE2 receptor was identified, like I said, in 2013. And this effort was led by a person called Peter Daszak. And he's been in the news as well because he was not only responsible for orchestrating the writing of that letter as soon as the pandemic broke, that this virus was not a lab leak, but was probably a zoonotic jump. So here he is in this paper published in Nature, where they identified SARS and that it used ACE2 to infect the cells. In this paper, he claims that an intermediate host is not needed for this virus to infect humans, and he touted for a pathogen discovery program. So this paper is actually contradicting what is now thought to be an accepted knowledge that there has to be an intermediate host. But in many papers, it's been proposed that SARS-CoV-2 don't need an intermediate host. So here again, they show that in many different cell types, on the left here, this virus can infect and or can be transfected and establish an infection. And here they're showing that it is via ACE2 receptor, which if they take HeLa cells that do not express ACE2 receptor, they do not find presence of the virus. So following that, there were other papers that showed that these viruses that are circulating in the bat, they have a potential for infecting humans. And here is again another sort of a gain of function mutation that they did, making chimeric viruses that were pretty pathogenic. And they, in this study, after infecting mice with this virus, they did not find that monoclonal antibodies or the vaccines could actually neutralize the virus. And again, the vaccines interestingly used were those that target the spike protein. So again, these people claim that there's a potential risk of SARS-CoV reemerging as which could infect the human population. So then there are others, including the past president of the Infectious Disease Society of America, David Relman, who made the statement that humans are fallible and lab accidents happen. They happen far more often than we care to admit. So if you, and of course, since then, there's been a push to reevaluate the origins of SARS. So if you look at the history of lab-acquired infections, as we all know, we work with bacteria, fungi, parasites, and viruses, all of which can be pathogenic to humans. And I'm just focusing on the viral infections, mostly lab-acquired or working in settings where you could acquire these infections. And it's like in the previous slide, it was said that these are not that rare. So, which is, in this case, when I was doing this research, it sort of surprised me that the HPV infections, the rate seems pretty high, 3 to 5 people per 1,000 workers in the US. And of course, there have been other viruses that have caused lab-related infections. So interestingly, the original SARS was also had infected a patient or a student in Singapore, a 27-year-old graduate student. And he had initially denied that there was any exposure or he could have gotten sick from that. But as you can see, this case report published in the New England Journal of Medicine, where despite denials, he had to be readmitted, and then it was found out that his sample actually tested positive for SARS-CoV, and it was exactly identical to the culture that he was working in the lab. And in fact, he had ended up cross-contaminating that vial with also a West Nile virus. And this concluded that this case is scientifically important because it documents lab-acquired SARS-CoV infection, and they were able to rule out any non-lab sources of infection. So pretty early on in this pandemic, although this sort of initiated or started in China much before the rest of the world realized, or even here in the Bay Area, we realized. But once that had happened, some of the earlier deaths that happened in 2019, they went back and tested whether those samples were positive for coronavirus, and indeed they found they were. So one question that's been sort of puzzling me is that now that we have all this information, now that we can go back and look at the virus, why do we not do that for our stored biospecimen and actually figure out when did this virus first emerge in the human population? So again, we have this doomsday scenario about, is this COVID-19 virus really very deadly? How much has it killed? What kind of havoc has it created? And if again you look through the noise versus signal, definitely the health status of an individual, the other comorbidities, they all sort of determine what the outcome would be. And it's not just for COVID. As we know that if anyone has more than one disease together, the chances of comorbidity, when you have two comorbid disease, the outcomes, health outcomes are not that great, and the chances of mortality increase. So there is a website which is a resource pool from WHO, CDC, the World Bank, and the United Nations, where you can actually visualize the number of deaths caused by different diseases in real time, and I guess since the start of the pandemic. So it's you can look at from March 2020 to now, and each day it'll give you that breakdown. So I had done this presentation in May, so I have this sort of historical data here, and as you can see, it tells you the number of COVID-related deaths, say in India. So COVID in May was not the #1 killer in India. You have more than about 318,000 deaths, number 12 in the number of people who are being killed because of COVID or dying. In Australia, there were only about 910 deaths, but the scenario is very different in the US and in the UK, where COVID was the #1 killer. But still, if you look at absolute numbers, you can't really compare because the population sizes are very different. So interestingly, in India, even when COVID-19 was being hyped, there were more people dying from influenza and pneumonia than there were from COVID-19. And of course, heart disease, the numbers are staggering. So if you compare from May to now, the number of deaths from COVID have obviously increased, but influenza and pneumonia, still the number of deaths caused by that are double the numbers caused by COVID-19. So if you just break it down to number of deaths, sort of an average per day, heart disease related deaths in India are over 4,000, influenza 1,600 and 1,500 for COVID. Compare that to US is 355 deaths, in UK 33.6 on an average, Canada 17, Australia 0.32. So hopefully that gives us some perspective of where we are with COVID-19 related deaths and the kind of draconian measures and disruptions that have that this pandemic has caused on people's mental health and other livelihoods and life. So the CDC had stopped collecting data on breakthrough infections once people started to get vaccinated in the US. So it's again a little difficult to make sense because now you have data that's skewed, but the Public Health of England continues to collect that data. And here are some numbers from their technical briefing. It says technical briefing 19, and number of cases listed as of July 19th. So Alpha variant, number of deaths caused, 4,000 about. So about 50% or 49% of the population was infected with Alpha, and 50% infected with Delta. And the number of deaths caused by Delta is 461. Now, to me, this does not seem like Delta is deadlier than Alpha, but it also could mean that we have learned how to better deal with the virus, so or with the disease or treatments. Therefore, the deaths are not as many as when initially Alpha was spreading. So again, we need to have a little bit of balance and sort of think rationally rather than jumping to conclusions. So Delta, so the number that I am not able to reconcile with is that in my previous slides, the data that was shown from aggregate from WHO and CDC seemed to say that there are about 127,000 deaths in the UK, but this is saying as of July 19th, the number of deaths that are shown are much less. So there is something that I have not been able to reconcile. If you look at the number of deaths caused by Delta in vaccinated versus non-vaccinated population, again, in the UK, as you can see here in this table, that they've divided that into people less than 50 years of age and greater than 50 years of age. So in the vaccinated population, the number of deaths caused by Delta variant under 50 is 4 versus 34 in unvaccinated, but the cases are also, numbers are different. So again, looking at these raw numbers might sort of give allow people to reach a different kind of conclusion, but if you were to just look at the percentages wise, the number of deaths in under 50, whether they're vaccinated or not vaccinated, does not differ. But where it does make a difference is in the older people, greater than 50, where in the vaccinated population, the number of deaths were much lower than in the unvaccinated population. But if you look overall, it still seems like the number of deaths in the unvaccinated population is less than in the vaccinated population. So we've obviously experienced this information overload, and the conclusion that I've reached is that for the general public to consume science in real time can be very confusing. So we are all used to sort of having the data change from time to time, or as more information comes, you sort of say that, okay, the past information was not entirely correct, and you're okay with that. So we are able to reconcile that and move on, but the general public sort of sticks to what is being told to them, and it's very hard for them to say, oh, but 2 months ago you told this, and now you're saying a completely different thing, or things that don't sort of match with what was told to us before. So, and of course, we've had issues in this country with our past president, who completely denied existence of COVID, even though he himself had COVID. So, and then whether or not symptomatic or asymptomatic people pass on this, or they can transmit or not transmit. And many experiments have been done recently, but again, if you go back to 1946, there was this paper that was published that was called the snot spattered experiment. And here a mathematician teamed up with a infectious disease a pathologist, and they were able to actually determine how far, when a person sneezes, do these particles spread. And they actually also had a load in those, meaning they were actually infectious, and they could then try and recover them and see how much of that actually matters in terms of clearance and things like that. And their conclusions were that if you're in a well-ventilated room, within 20 minutes, this is all cleared up, and it's very difficult to get infected. So if you're outside, for me, wearing mask makes no sense. But if you're inside in a crowded place, of course, mask makes sense. So nonetheless, in August of last year, we were at a stage where they were able to find that the viral load in some of the asymptomatic people were similar to that in the symptomatic patients. And with that, they reached the conclusion that asymptomatic people can also spread or can transmit the virus. Now, if you're asymptomatic and you remain asymptomatic, I'm not sure how that makes sense, but if you're asymptomatic, which we are all at the beginning of any infection, then of course you can, and you become symptomatic, then you can transmit. So, and then just last week, it was reported by CDC that in Cape Cod, a county in Massachusetts, are there any questions? Sorry, are there any questions or?
I don't think there are any questions. I think someone's just like logging in and logging out. So
Okay.
Okay. Maybe I can ask Aditi.
Sure.
So from what I could gather, you're saying that the lab leak possibility is the strongest possibility than the natural
I think
origin from
the
rats.
jury is still out. I think they had enough publications to say that perhaps you don't need an intermediate host. And the question remains as to how does this virus, I'll address some of that again. How did this virus acquire certain signatures? For example, ACE2, and then the virus has certain furin sites that are not found in any other animals except for humans. So how does that furin site come about? And we can go over that. So just going back to the recent findings that were from Cape Cod here in the U.S. was that in all the infections that happened, 74% of them occurred in fully vaccinated people. But more importantly, what they found was that the viral load or the cycle threshold for RT-PCR was pretty similar between the fully vaccinated and unvaccinated. So again, that would say that the amount of virus present in the vaccinated, fully vaccinated, and the unvaccinated is exactly identical. So if that's the case, then what's the rationale for saying that the fully vaccinated people cannot spread the virus? I do not understand that. If they have the same viral load, they have symptoms, then they're just as infectious as people who are not vaccinated. So going through the publications that have on COVID-19, to me, some of these publications really defy our sort of known publication standards. And it's up to the scientific community to decide how much trust they place in that. Are they robust? Are they reproducible? The jury is still out. So if you just do a PubMed search on SARS, as you can see, between 1911 and 2003, there were just over 1,800 publications. Between 2003, when the first SARS pandemic appeared, and 2021, there were 31,600 publications. But just between March of or December of 2019 to October of 2020, the number of publications literally doubled. So we had 39,000 publications. And now between October and August, we have a total of 65,000 publications on SARS. So it's really hard to make, nobody can keep up with all those publications. So where are we with, just to remind you that when the first SARS epidemic broke, it took them over 10 years to find the virus. But here we are, supposedly the pandemic happened in November, and come January, we had this publication where they said that this outbreak was associated with a new coronavirus of bat origin. So this was published in Nature. The paper was received on January 20th, accepted for publication on January 29th. They had isolated this virus from a patient in China, and they reported that it was 96% identical to a known bat coronavirus. And soon after that, there was another publication on the crystal structure of the receptor binding domain of SARS-CoV-2. So remember, the outbreak happened in November, and here we are, paper submitted for publication on March 3rd, accepted for publication on March 6th on crystal structure of the receptor binding domain of coronavirus or COVID-2. You contrast that to another crystal structure paper also submitted for publication, and it took over 6 months before it was accepted. So going back to this epidemic of or pandemic of COVID-2, so this is a pretty exhaustive review article published more recently. And here's a depiction of perhaps how this could have happened. And the question comes about, again, this intermediate animal host. And yet you've seen publications from Peter Dasnek's lab that claims, and other labs that claim, that we don't need an intermediate host, that this virus is capable of emerging into the human population. So why do we need to look for an intermediate host is not quite clear, because again, it was being touted that we don't need one. So that comes back to lab versus natural jump. And again, some of the points that have been made is that this COVID-2 has this polybasic furin-like cleavage site. And again, Dr. Kannan and his expertise, all the chemists over there, could perhaps have more to say about this furin-like polybasic stretch that has appeared in this virus, which is only found in human signatures. So that remains a mystery. Nonetheless, in some people, as we can see, that it causes severe respiratory distress and then ultimately death. But we do have people in which there are no symptoms, and as actually we do know that bats, even though they have infection, they actually do not have any clinical disease or symptoms. So the infection clears by itself. So similarly, in humans, there is a population or a subgroup of people who can get infected, but they remain asymptomatic. And then there are people with mild symptoms, and then there are people with severe symptoms in which it can lead to death. So in just doing some preclinical study, here is one paper where they had infected these macaques with SARS isolated from a patient who had traveled to China. And again, they say even though they gave these monkeys a bolus of the virus, none of them actually developed overt clinical signs of the disease. So in terms of postulates, I'm not sure if it completely satisfies that, but again, we have some observations here. And to note that out of 4 monkeys per age group to young and old. Only 2 from each age group actually had any viral load. So now they're showing data from only 2 monkeys per age group. And here is from nasal swab and from throat swab. And this is how much they detected. And here is in all different tissue types. So there is a lot of variability, as you can see. And even though they call it viral shedding, when I wrote to the authors asking them if this is all from the swabs, but whether they were able to grow the virus, even from the swabs, there was no response. And it's been very difficult to even grow the virus in culture, but actually, as it was noted in the initial discovery of the coronaviruses by Dr. June Almeida, that these viruses are pretty hard to grow in culture. But it takes 2 to tango, so the person who's infectious can give it to another person, and that really depends on your own immune system, your own other health conditions, whether or not the infection is successful in establishing itself. So again, some publications about the mRNA vaccines, and this was a review published in 2018, a lot of excitement about the mRNA vaccines. And as you can see, the list of vaccines that were in clinical trial, a lot of them on HIV, some on Zika, on influenza virus from Moderna. And as of 2018, it's still listed as ongoing and recruiting. And when you look at on clinicaltrials.gov, you can see that even though the trial finished, there were no results posted in here. But they had a link to a paper that they published. And if you look at that paper that they published, it was terminated as a phase 1 trial. It only went on for 43 days. And it was in about 200 and some people. But their intradermal vaccination was terminated because of several severe side effects. And in the intramuscular arm, they list here over 124 unsolicited adverse events, which are, some of them are highlighted here. So it remains a mystery, at least to me, what happened between just May of 2019, where we only had a phase 1 trial, to fast forward 2020, where this is a complete success. So when you look at the documents submitted by Pfizer and Moderna to various agencies such as the FDA, the European Medical Association, and the Japanese authorities, and here are some data from the biodistribution studies. The number of animals to me are extremely small, and in a 4 here, it's most of the document, I mean, it's hundreds of pages of document. They're available, anyone can look at it. So here is the distribution study in 4 rats. And then if you look at pharmacokinetics, again, the number of animals per group to me is pretty small. I'm not sure what statistical analysis can be done or what conclusions can be reached from an N of 3, but here we are. Moderna was a little bit better with 5 animals instead of 3 per time point, but that's what we have here. So in their documents that we submitted to the FDA, between phase 1 and phase 3, it was listed that we had 44,000 participants. But when the data were presented, it wasn't really, in terms of when you're looking at either T-cell responses or neutralization effects, the N's were pretty small. So what happens between 44,000 and how does it come down to 10, 15 is not quite clear to me. So this was the timeline that was proposed, that they would follow up for 2 years after vaccination. But again, that was not quite what happened. Pretty soon, within 2 months of giving the first dose of vaccine, the whole placebo arm was folded into the vaccine arm, and so the follow-ups have been completely eliminated. So I'm not sure how do you evaluate the safety and efficacy when none of these the protocols that were put into the clinical trials are not being followed. And here was their primary efficacy endpoint listed, which is to decrease incidence. But as we can see, as the number of people who are being vaccinated are rising, so are the breakthrough infections. So whether or not it's decreasing incidence is again something that remains questionable. So we know that in the past, FDA has published this document where they highlight 22 cases where phase 2 and phase 3 trials had divergent results. And you can go and read this document if you like. I have just highlighted one case which talks about vaccines, HSV2 vaccine, where they said that after phase 2, even despite positive biomarker results, the phase 3 trials, it did not prevent herpes. So we don't even have really good data from phase 2 and phase 3 trials from these vaccines, and yet here we are pushing for a full approval and wanting to vaccinate everybody. So one thing to keep in mind is that if you do vaccinate every single person, where is your control group? How do you even make sense of whether or not, if you have any adverse event, is that caused by the vaccine or is it something that's happening naturally? So to me, that goes completely contradictory to my scientific training, that you have no comparison groups or no placebo without having any long-term studies. So what's happening is that the research findings that are, this is again a study that was done recently, and they have this headline where research findings that are probably wrong are cited far more than robust ones. And here's the paper. So to the students, I would just emphasize that read the original papers, look at the data and reach your own conclusions. And this is what I was told when I was doing my MSc, and we used to have this journal club paper. All my professors, Professor Modak, Professor Devaker, Dr. Kannan, all of them emphasized that look at the raw results and then see if the conclusions that you reach are the conclusions that the authors have reached, or is it that you want to make something look more interesting and more sexy than what it actually is. So looking at the SARS variants, they definitely seem to be emerging at a much faster rate than, let's say, the flu or influenza virus. And then, of course, this has also been of interest to me as to why is it called that the Delta variant as an Indian variant. So let's look at the evidence. So this is a slide taken from the CDC website where they talk about multiple variants circulating globally. And of course, as they say, that viruses constantly change through mutation, and so new variants are expected. And once they emerge, some of them disappear, some of them persist. And they're, of course, interested in the variants that spread more easily from people to people. And then some of these variants can have milder symptoms, some much worse symptoms. So the dreaded Delta variant. Now, this is a paper published in August of 2020 in Cell, where they look at these two variants. And here is your Delta variant, which is G614. And as you can see, which is represented in blue, and the original alpha variant, D614 in orange. And in February, obviously, the alpha variant was pretty predominant. But as you can see, that it's been present in the population from the very beginning. And as the infections established, you can see the Delta variant taking over. And in this paper, they already said that the Delta variant is more transmissible. And here is what they're showing, that before March 1st, you can see orange being the alpha and blue being the Delta. And here we are in India, and here is in Europe and America. The Delta variant is already the predominant variant in Europe. And here we are in March of 2020, and the Delta variant is emerging in India. So I remain confused as to why this is labeled as the Indian variant. Perhaps somebody else can elaborate on that. And this mutation is in the hinge region, but they also find that this mutation is actually accompanied by 2 other mutations, one in the 5' UTR and another downstream, including in the RNA polymerase region, which allows it to actually replicate much faster than the other variant. So I'll skip through some of these. These are all listed, the different kinds of variants that are circulating right now. And I think in the previous talk, Eddie had actually touched upon those variants. So again, the number of variants that are circulating, at least in the US, they seem to be pretty large. And given that this virus has only been around for a year, it's pretty amazing that the number of variants that have emerged. So again, like I said, it remains a mystery to me why the mainstream media as well as scientific publications continue to call the Delta variant as first discovered in India. Perhaps, like I said, somebody else could tell me as to why that is. So I would like to now touch upon the natural immunity versus vaccine-induced immunity. So vaccines, again, this is something that Eddie had touched upon in his talk last time, so I won't spend too much time, but you can have vaccines against viruses and bacteria, DNA or RNA viruses, and some of those are listed here. Now, I do want to spend just a couple of minutes talking about how we get to live attenuated vaccines. And that would sort of also answer the question that was asked about whether or not these viruses require an intermediate host. So we have live attenuated viruses, vaccine for a number of viruses listed here: measles, mumps, rubella, chickenpox. And the way that's done is that you take the virus, which is human virus, and you grow them in something else, in most cases and not in chicken eggs. And as you passage through them and have multiple rounds of replication, the virus loses its ability to replicate in human cells and now acquires the ability to actually replicate in chicken cells. Now, if this wasn't a controlled experiment, perhaps it could infect chicken population and wipe it out. But of course, this is done in a very controlled environment. And now you take that attenuated virus, it's still live, and put it back into humans. And because it's lost its ability to replicate in humans, but it evokes a robust immune response, and you have a nice, good vaccine. Similarly, you can take the virus and you can either heat inactivate and kill it, or you can chemically inactivate and kill it and then give it to humans, and you get a vaccine. And then there were all other kinds, which are recombinant vaccines in some cases with adenoviruses, and those have their pros and cons. But as we all know, the viruses never, when you have a natural infection, you have different components of the immune response that get activated. But with a vaccine, it's never always the full complement. So we've known that vaccines against DNA viruses are far more effective than those for RNA viruses. And here's an example of chickenpox. But we also know that somebody who naturally gets chickenpox, the chances of them getting it again are nil, except if you got it when you were younger than 6 months old, or if your infection was very, very mild, or you had a subclinical infection. The chickenpox vaccines are very effective, and they have done a great deal in reducing the spread and of the disease. But even with their effectiveness, you do see breakthrough cases. So again, this was covered last time by Eddie, so I will not dwell upon this, but so far for the SARS-CoV-2 vaccine, at least in the US, we have 4 vaccines which are under emergency use authorization, or maybe only 3. I'm not sure whether AstraZeneca has made it here or not yet. So we have these, again, from the slide from the CDC website, which talks about protection, vaccine-induced antibody and protection. And as you can see that they talk about whether or not, if you have a mutation in the spike protein, whether or not it can be effective or whether you have an escape. So the threshold for protection has not been yet determined, and the duration of immunity provided by the vaccine still remains questionable. So very quickly to talk about the immune system, as we know, it is very important in keeping infections at bay. And we have a number of different components of the immune system, which I equate to all these different parts of an army. And we need all of them working to be able to win the war. And there is, even though the vaccines have been touted as the magic potion, I'm not quite sure whether they are in all cases. So our immune organs, we have several of them listed here. Some of them are very key to our proper functioning of the immune system. So the bone marrow, of course, makes all our progenitor immune cells listed here of myeloid origin or of lymphoid origin. So again, a lot of you may be familiar with these, that we have these different kinds of cell types, neutrophils and platelets, which are the first line of defense. And almost in any kind of infection, bacterial or viral or even injury, neutrophils are perhaps the first ones that show up at the site of injury or inflammation. And they are very important for phagocytosis and part of our innate immune system. And then you have the T and the B cells that need to be trained before they become part of your adaptive immune system. So we have our scouts and camp builders that are part of our innate immune system. So the T cells, before they are of any use, they go through this process of thymic education, which is extremely important. And these naive T cells from the bone marrow, they travel to the thymus, where these naive T cells start expressing certain receptors on their cells, the CD4, CD8 kind. And they are in the outer cortex of the thymus. They are first presented with a non-self signal. And this is their first test, where if they don't identify that as a non-self and kill that cell, then they themselves are killed. But once they pass that test, they can go to the next inner cortex, where they are actually presented with something which is called self-antigen. And again, if they don't recognize this as a self-antigen, and it is important because if they don't recognize this as self-antigen, we get what is called an autoimmune disease. And if it recognizes that self-antigen and this handshake is not very tight, then it passes on to the next step, where in the inner thymus, they then divide and become either T helper or T cytotoxic cells. And again, they go through several training steps. And if they fail, they're killed. if they pass, then they are further subclassified into different kinds of Th1 to 17 Treg cells and the T cytotoxic cells. And then comes the second part of our training, which is before they can make clones and several antibodies, which happens in the secondary lymphoid tissues, such as the lymph nodes and the spleen. So where, of course, other players come into action as well, which is the cytokines and the interleukins. So it's a very complex system, and really, most importantly, thymic education, which is really important for our adaptive immune responses. As we can see here, as we age, the number of cells that the thymus outputs actually decreases exponentially. So whether or not this mRNA can induce both adaptive and innate immune responses remains questionable. It's assuming that the mRNA that's given is never degraded, or we don't have any RNA population without the cap or the poly-A tail, or we don't have any truncated proteins. So if that's the scenario, then hunky-dory, you get spike protein, you get antibodies to certain epitopes, and you have a good, robust adaptive immune response. But it's hard to imagine that all of that RNA, none of that escapes any degradation, partial or full degradation, which might evoke. Can we have someone mute there? Okay, so natural immunity then, when you have a natural infection, you will probably make antibodies against several components of the virus, which is not just the spike protein, but to the nucleocapsid protein, to other envelope proteins, and perhaps even to the RNA itself. Whereas with the vaccine-induced immunity, at least in this case, it'll be only against the spike protein. So in my opinion, you need a full set of components to be able to combat or win that war, rather than just a subset of that. So we have a recent publication where they actually looked at, again, just the spike protein neutralizing antibodies from naturally infected people versus people with the vaccine, where, again, the N is pretty small. And they, for the spike protein, they can see very comparable neutralization. And they also see some B cell division, making them conclude that the vaccinated people, as well as people with natural infections, have certain long-term memory against the virus. So just very quickly, again, the way we've been primed to think that if you have any adverse events or infection or reactions from vaccine, in this case, in this person, he said that he had chills and he shook so hard that he cracked a tooth after taking his second dose, that's acceptable. But if you get chills and fevers from natural infections, somehow that's not acceptable. But really, it's amazing that we not even have results from phase 3 trial, and here we are asking for a full approval. So I think there are also some questions about whether COVID is long haul or not, and can you have those symptoms? But again, it's not unique to COVID. There are many other infections and diseases that cause long haul symptoms. Those include from Ebola to strep throat, and from chikungunya to dengue, as we know in India. So there is also, of course, the other side of this whole pandemic that we haven't looked at, which is the mental toll, which is really hard to determine. But more and more data is coming out that the mental toll that it's taken on people, that is really unprecedented. And that it is a silent killer. You cannot quantify in any way the mental toll it's taken on our children, on our elder people who spent time in isolation, and a lot of them died in isolation. So again, we got into that because of some of the work that we were trying to assess the mental toll on people during this pandemic. And that data is not quite published, but what we found is that contrary to our thinking, people who actually had pre-existing mental disorders such as PTSD, post-traumatic stress disorders, they actually did better during the pandemic because they didn't feel so isolated. They didn't feel the social pressure to go out and socialize. And so they actually fared better than we thought. And whereas the normal population actually didn't. So what does stress do to you? And stress does to your thymus, just like the thymus as we age shrinks. We find that in animals, when they are either stressed acutely or chronically, in both cases, both the thymus and the spleen shrinks, and the number of cells that they put out also decrease considerably. So that was in chronic stress, acute stress and chronic stress as well. You also change the B to T lymphocyte ratio, and that correlates with the cortisol levels. And not just that, in animal models, it also correlates with the anxiety-like symptoms as well as depression-like symptoms. So again, acute or chronic stress can result in your compromising your thymic function and therefore your immune function per se. And so in conclusion, again, this is also a conclusion reached by CDC that it's difficult to estimate how results from these studies will translate to clinical protection in case of vaccines, and that to keep in mind that these neutralization assays are somewhat artificial. They deal with non-replicating or pseudoviruses, and the use of limited or full sets of spike mutations obviously compromises where we stand with what we understand. And most importantly, that most of these published studies have small sample sizes, and therefore you cannot generalize. But again, we are here generalizing all the data. So with that, I will just stop, and sorry I've gone over a little bit, but I'm ready for questions and answers.
Please unmute. Nitika, unmute.
Yeah, can you
Yeah, we can hear
hear
you
me
now.
now?
Yeah.
Okay. So thank you, Professor Aditi. You gave us a very insightful talk on COVID virus. You touched on many aspects of the virus, how it emerged, questions related to how it emerged out, then the fact whether mRNA vaccines, whether they are indeed working or not. And a very important valid point that you raised was that if we do mass vaccinate, are we going to lose our control population? So all the AEs that we're looking at, the adverse events that we're looking at also, in the clinical studies, assuming we mass vaccinate, where will our controls be, and whether it's a COVID related stuff or, it's the background of the noise that we're considering. That's a very, very valid point that you've touched upon. So I open the floor to questions. We do have a question in the chat box. If you could take the question first. Can you read the chat box, or should I read out the question to you?
The chat box question is a bit long, so I let others raise. In the meantime, she can respond to that.
Okay. So we have Dr. Utpal with a question.
Yeah, thank you, Aditi. Can I come in?
Yeah.
Yeah, thank you. Let me first of all congratulate Professor Bhargava for such an extensive coverage of certain burning scientific as well as social medical topic, which is affecting the entire world currently. And I think it's an excellent choice of lecture topic by Professor Kanan. I also thank him for this arrangement. Okay, let me go to my questions. First of all, Professor Kanan and me, and probably he also discussing with others regarding the, innovations and scientific research related to COVID. And you have presented one slide, a discussion point regarding the quality of the research publications. And it's really alarming.
Can I
Now,
request
this is a
everyone
linkage
else
with
to mute themselves?
Sohan, can you mute? Can you mute yourself, Sohan?
Yeah, there's some kind of disturbance. Yeah. Thank you. Okay.
I've
So okay,
muted
let
you.
me be brief. Am I audible? Yeah, am I audible?
You are audible. Yeah, you are
Yeah,
audible.
thank you, sir. Yeah. So so let me cut short. Right. So there's a hello.
Yes, Professor Bora, you are audible. Please
You're
carry on.
audible.
Now you are not.
I think there's a technical glitch there also. So maybe I can ask you a question, Professor Aditi, in the meantime. Sorry for butting in like that. So you do work with a lot of patients who have chronic inflammatory diseases also. And these individuals are constantly treated with these immunosuppressive drugs. So what is your take on it, that if in case patients who are down with chronic inflammatory diseases, they are on these medications, how will these people respond to vaccination if done, number one? And God forbid, if they come down with COVID-19 infection, what is the line of treatment which is used for such patients?
Thanks, Nikita, for that question. I mean, I'm not a physician. I don't really treat patients. So I can only answer in terms of, I mean, I think part of the problem that we had initially in case when this pandemic hit, that we had such large number of deaths is because there was a lot of confusion as to how to treat these patients. And so, for example, I do know that of a few cases where they went in after their first dose of the vaccine and they got COVID-19 infection, and they were then given steroids, and they didn't realize that these people were diabetic, so their blood sugars shot through the roof, and they actually died because of blood sugar going up, and not so much because they had COVID infection. So there's a lot of, we're not quite clear. I think that's something that we're still learning as to what are the kind of treatments that need to be given. And there is no one treatment that fits all. So like I mentioned, people with different comorbidities, it's really important right now to understand how those comorbidities affect, and for your physician to know what comorbidities you have. So especially if you're diabetic or if you have other metabolic syndromes, that's something to talk about when you go in with COVID diagnosis.
True, and as Dr. Raghuram has mentioned in the chat box, there is, I mean, indeed it's a question whether it's treatment or just management of symptoms that we're doing at the moment. So another point to look at. So we have another question in the chat box. If you could take that, please.
Would you read it? That
Sure.
might be easier.
So this is from Dr. Narayanan, and Dr. Narayanan asks that are there any insights on IIT Delhi researchers who claim they were the first to report on the 4 insertions in spike glycoproteins, which are unique only to SARS-CoV-19 and not present in any other coronavirus? The sequence of amino acid residues inserted was similar to HIV-1 DP120 and GAG. Apparently, it is learned the researchers were forced to withdraw from publicizing the data. So that's the question from Dr. Narayanan.
It's difficult for me to comment. I haven't seen that paper. But again, as we do know, people who work with cloning, that you can clone certain things, and then when you put them in cell culture, they will spit it out because they don't like it. So it's whether or not there've been many publications you can go where they've mutated many residues in the virus, or they might have put in something in there, and it's hard to say what remains and what doesn't remain. It's also hard to say once it goes out in or you put it in vivo, how will the virus behave? So I haven't seen the publication. I do not know whether or not they found signatures of HIV in it or not, but there have been many other full sequences, and that kind of forensic is something that should be done, definitely should be done. So my mother has actually joined in, and she has a question, but I can't unmute her. So,
Karan sir, I think you will have to unmute because you are the host.
Yeah, I'll just do it. But in the meantime, let's see. Lakshmi, yeah.
Are there any other questions in the chat box?
Yeah. So
Dr. Raghuram has a question. Is there any treatment at all, or is it just management of symptoms that we're talking about?
Well, I mean, I definitely think in terms of treatment, there are many indications that have been used.
Can you please mute yourself?
Sohan, can you unmute? Yeah.
I muted him.
Yeah, so there's been so much of politics around that, with, let's say, with ivermectin, with remdesivir. At least according to the WHO trials, remdesivir did not have any effect compared to placebo. In terms of vaccines, I don't think that there are vaccines. For example, if you just take the virus and you inactivate that virus, and I think there's a vaccine version for that, the Indian version, which is a long tested technology. Would that be effective? Probably. And again, the jury is still out for the mRNA vaccine. It's not that it's completely ineffective. At least it suggests that it is. And as far as other treatments, again, it really depends on the person's health status. And so if you have diabetes as a comorbidity, treating with steroid may not be the wisest choice. But if you're otherwise healthy, then again, and that's up to the physician to decide.
Do we have him? I think Modak sir has a question. Sir, please go ahead. Sir, we can't hear you, sir.
So I just saw that vaccines are not really stopping transmission. There's enough evidence based from many vaccines that they don't stop transmission.
Can you hear me?
Yeah, we can hear you. Yes. We can
Okay.
hear you.
Well, the spike protein of COVID-19 contains at least 12 most unusual pentapeptides, not found in any SARS family of proteins. Now, this is very unusual feature that you may worry about because that makes COVID-19 a very different one, so much so that as if it looks, it has been engineered. And some of these peptides are so unusual, some of them were called as forbidden pentapeptides by some people. Obviously, being present, they are certainly very rare and they have been present in COVID spike. So this is something one must worry about when one condemns
Sohan, can you
any
close your
viral
TV? Your TV noise is coming. Sohan, we can't hear you. Your TV
I'm
noise
sorry,
is coming.
I'm in a place where it's very noisy, unfortunately.
Yeah, so we are not able to hear you.
I heard that there are unusual pentapeptides in the spike of COVID, and again, like I showed from those papers, the SARS-CoV virus has been infected in many different human cell types. Now, as we saw that it can acquire features from those cell types, and whether that gets integrated and then you see it in what has emerged now, that's not something unusual.
Yeah. I think it's actually the peptide map of entire COVID-19 protein is very unusual, very unusual, very different. Uh, in fact, it's so different that it's shocking that people still consider as a family of SARS-2 family. Because it looks similar, but it isn't. It isn't at all. I wonder to what extent people have considered, although people in popular terms, people say some people may have engineered it, it may well be, but actually the nature of engineering has not been really revealed so far. And we're trying to do that, I must tell you, and maybe we'll let you know in a month or so. It's dramatically different. So therefore, any attempts to make vaccines or let's say antibodies to these proteins will be important because they will not resemble any kind of immunity that has been acquired by against SARS-2, for example, or similar family of proteins. Although I do agree that a consistent or constant challenge to the immune system by variety of antigens or vaccines will generate normally a facilitation type of response to any new kind of challenge. I agree and I understand that. But this is very unusual that you must look into.
Thank you for your comment. I think people are looking into the spike protein variability. So we have a question from Nitin about adenovirus-based vaccines and that they may not work for the Indian population. So, Dr. Moda, can you mute yourself because of the background noise? Yeah, thanks. So again, it's hard to say whether or not the adenovirus-based vaccine will work or not. The other thing to keep in mind is that the adenovirus, even though it's not supposed to integrate because they've taken out that portion, whether or not it integrates. And I mean, I've seen papers from Indian publications which have looked at antibodies being made after, is it now Covishield? That's the adenovirus-based vaccine that's given, that there are antibodies that are made. So, but how effective they are, again, to the real reinfection, that's the test. Whether or not they can neutralize a spike antibody made in the lab or from a pseudovirus is what is normally published in paper, but that does not equate whether or not when you have a natural infection, how effective that will be. And the fact that we hear of breakthrough infections only in fully vaccinated people, but we don't really hear of breakthrough infection in people with natural infections is something to think about. And so again, if the rationale for vaccinating naturally infected people is a puzzle, and why are you, again, like I said, we need a control group. We also need a control group of unvaccinated individuals that have natural COVID to see which is more effective or which gives you a longer term immunity.
I think Nitin has a question.
Yes.
No.
Nitin,
Yeah.
is already a rest.
Yeah, this was the one.
Yeah.
Yes. Utpal has a question.
Yeah, Dr. Utpal, I think, wants to ask.
Thank you. Sorry, actually, I got disconnected for some reason. I'm unable to understand. Yeah, okay. So I think one query I was trying to ask you has already been discussed. I was listening to others, right? So I will straight away go to another next one. So you were speaking about some breakthrough infections, and now you just told that people who get naturally infected, they are not much susceptible to breakthrough infections. But vaccinated people probably are more prone to breakthrough infections. India, the number of cases of breakthrough infections are really very high currently. Now, is there any kind of study which is trying to find out whether it's the alpha 1 or the delta 1? I see these are the two most dominating variants. So which one is actually having an upper hand in the breakthrough infection cases?
It seems like that right now it's the Delta variant that is more predominant, but I think CDC is, and in the recent breakthrough cases that they sequenced from the Cape Cod event, it's the Delta variant. So it's been established that Delta variant it has a replication advantage. Like I mentioned, it's not just a mutation in the spike protein, it's accompanied by two other mutations, and one of them is in the polymerase gene, which allows it to make more copies. So again, I think it's in the virus's advantage to have a mutation that will allow it to spread, but not have it kill its host. Because if it kills its host, what's the point?
Okay, thank you. So just my next question. So you were discussing about the quality of publications. So and obviously there is also a lot of doubts being raised about the quality of scientific work being done in such a hurried time, and mostly when the laboratories were closed. You don't have really research workers visiting the labs, and many labs have come out with some scientific discoveries which are really doubtful because the labs were closed most of the times. So there is one real concept. But now,
well, I wouldn't say that. So I just want to correct
speaking about
you, Paul, that at least here.
The Indian scenario. I'm telling you about the Indian scenario. I have to give you the reference.
Yes.
I'm not telling about the global scenario. In India, most of the labs are still closed, and they are not functioning, to their full potential. And it's really doubtful how these research were done. Anyway, now thing is that there are several kinds of actually pandemics going on parallelly. COVID is one pandemic which everyone is aware of. You told about another pandemic, that's the mental health disease. But you told that people with mental health disease, histories of mental health disease, in fact, were doing better. But probably, a new, group of people got added to this mental health disease who were perfectly normal before the pandemic. Now, amongst these, there is a kind of a tendency among the scientific community to rush and publish and kind of gain some kind of glory. And it's something they're trying to do, like make hay while the sun shines. So that's also one kind of a pandemic, as I can see it. Now, this is actually kind of contributing towards the other pandemic, which we are not referring to as infodemic. And policymakers and the media rely heavily on this kind of information is generated in the laboratory, which is being passed on to the public. Now, I think what could be the approach to really stop this? This is serious kind of a malaise. Yeah, that's my discussion point.
You raise very good points, and that's definitely gotten worse with the pandemic. Like I pointed out, there are so many publications. Now, that doesn't mean that all publications are not robust or not reproducible. Again, you need to take a look and see. And it's very difficult to go through 60,000 publications. You can't really do that in a objective manner. So you can look at the ones that you're interested in and see how robust they are. And it doesn't mean just because someone comes from one lab versus the other that they will be less robust or more robust, in my opinion. But like you're saying, that the mental health consequences of this pandemic, it's very difficult to determine that. I mean, these are very subjective way of assessing, right? So people, the way they would answer the questions, we also find just generally, even when you send out questionnaire-based surveys, women tend to get back to you more often than men. So as it is, the population gets skewed. So when that happens, then you say that the prevalence of this is more in women, which is not correct. Again, because if men are not giving you back the surveys, your ends are much smaller. So there are caveats to any of the studies that you do, and as long as you're aware of those caveats, I think, and you state them, then I think that's a good approach.
Yeah, thank you.
Thank you, Dr. Utpal, for your questions and discussion points. We have another question from Dr. Raghuram there in the chat box about the testing for T-cell immune responses against the current vaccines.
Right. So I haven't exhaustively looked at the literature, but when I try to find anything that's at least for the mRNA vaccine, they're only focusing on B-cell immunity. Like I said, in one of the review articles that I cited, they did try to look at it, but I don't think, like I said, I haven't seen any in vivo data to say the spike protein, full-length spike protein is being made, or there is no degradation of mRNA and things like that. So it's hard to say whether or not they're full complement or what are the other components that are being investigated, but I suspect that will happen.
Eddie, I was waiting for you to give your comments. Eddie, are you there?
Yeah, I'm there. Aditi, firstly, great talk. Absolutely great talk. It is really important for people to not get swayed by the mob mentality. It's important for people to actually understand the science and give different perspectives. What we have learned in this pandemic is we are still learning. There's no right or wrong answer. There's so many lessons to be learned. And I think over a period of time, we will learn how to handle this better. If you see the number of deaths that occurred initially in the pandemic, and today when you have more data and you look at it, you know that it was mishandled. The doctors could have handled the patients better. So now we have better methods in terms of how to handle the patients, what needs to be done. So it's a learning lesson even in the hospitals. the doctors. I'm from a vaccine field, so I can tell you one thing definite, is we used all the different platforms that can be used for making vaccines. And what we need is a rapid response. The traditional vaccine platforms could take about 2 years to get a vaccine, and it's too late in a pandemic. So one thing that we have found, which is surprising, is mRNA did not work for any of the other diseases. I worked on HIV, mRNA, it didn't work. Rabies, and as well as hepatitis B. We got good animal data, but in human it wasn't good. And I mentioned that we got lucky in the case of COVID-2. So what's actually going to happen is from here on is whenever you have a pandemic, an immediate response will occur with an mRNA. You will be able to buy some time, get people immunized, generate some type of immunity, but then the other platforms will consolidate the immune response. Let's not give up on live attenuated vaccines. Let's not give up on inactivated vaccines. I'm a firm believer that a human response generated to a natural infection is far more robust than any of the platforms. But you cannot wait for the population to generate an infection and generate an immune response. You have to have a prophylactic approach. So what is going to happen is you're going to have the mRNA vaccines coming in first. You're going to have some protection, and then the other platforms will cover. I have learned a lot of lessons, and I continue to learn lessons. So I would accept the science in totality. And one question I want to respond to Utpal, is during this pandemic, what nice has happened is something called preprints. Earlier, it used to take a very long time for the publications to come through, and there was information that was not available to the field. But with preprints, what you're forcing each of the PIs is to present the data. So the data is there. Let's not get swayed by how you interpret the data. It is up to us. I think the information should be available. Let's not prevent the preprints from happening. So, just a few comments. I still feel we don't know the right or the wrong answer. We have to take it, as it comes.
So thanks, Eddie. But I mean, I feel like you're saying that the mRNA efficacy is already a foregone conclusion. I don't think so. I don't think you can say that we got lucky. It has it worked in probably mitigating symptoms in the older population? Perhaps, but has it worked in the younger generation? We don't know. There is definitely in young adults, especially males under 30, the number of myocarditis and pericarditis cases that have happened because of the mRNA vaccines is pretty high. So to completely ignore those and say that the pros outweigh the cons is not correct. It may outweigh in the older population over 65, but not so in the younger population. Like I showed you the data, vaccines are not preventing deaths. The death rate is exactly identical in under 50 for vaccinated or unvaccinated. So I don't really think that it's a foregone conclusion that the mRNA vaccine for COVID-2 is worked and we got lucky. I mean, I think you can get lucky with discoveries, but you need to show the data. And I would like to see the data without the noise to be, and I would like that. I mean, the idea of RNA interference and RNA platforms is I think pretty attractive, but you still need data with proper controls. One thing that none of these studies have is a non-specific RNA. Give that non-specific RNA and show that you're not getting similar kind of protection from evoking an innate immune response. So we don't have that data. So then to make that conclusion that this mRNA is specific and working remains to be seen. We don't have that data.
Yeah, Aditi, as we always say, the jury is out. We are open to interpretations. Let's take a pandemic X that may come tomorrow. Okay, So we'll have a choice not to do anything till the best vaccines work, and it could take 2 years, or you can do a plug and play and at least generate some immune response and hope that the people in the vaccinated group, there's lower mortality in that particular group. So we'll have a choice to do. And what we have clearly seen with the mRNA vaccines is, after the 2 doses, we are seeing antibody titers similar to the covalescent serum, which is a titer of 100. Now, I don't believe that that would have been generated non-specifically. It's a very specific response. So it's very clear that you are generating a neutralizing antibody similar to covalescent serum. And in a pandemic situation, you need that. Otherwise, people are going to die. So my view is, in the initial stages, you need something really quick. And if a particular platform can deliver that, I'm ready to take it.
Right. So again, I think I still think that in the convalescent serum, you get antibodies to nucleocapsid, you get to many other proteins that you're not looking at. And because you are just focusing on comparing the antibodies against spike protein, because you don't have any other for the vaccinated group, and you make your conclusions, then I think they're half-baked conclusions. So definitely, I agree that you target the more vulnerable population, but that does not mean you forcibly vaccine every single person, because the jury is still out about the adverse events. We still need people. We still need to understand what are the adverse events that are happening, and there are some serious adverse events. There are people who they have other comorbidities in which there is cross reaction. And to just wave that off to say that, it needs to be people's choice. You make the choice knowing that people, if you go out and drive on the road, that you can get hit by a driver who's not as good, but you have that choice. Nobody forces you to go and not drive, or nobody, and one point that I would like to make is that, however harsh it may sound, but there is Darwin's natural selection, right? So with this pandemic, you're seeing people who are sicker, and unfortunately, globally, people have gotten more health problems. That the pandemic is taking out, or at least killing, people who are sick. But we consciously make a choice of sending our fittest to fight wars, and we kill them. And then what? So when you talk about social and moral responsibilities, it doesn't end there. So before we make these kind of judgment on people, I think we have to be careful as to what are we, where are we going with this?
I just have one question. You said, the thymus gets shrunk over a period of time. Mm-hmm. And in India and all, first we started.
Kanan sir, you're breaking. We can't hear you, Kanan sir. Yes.
Okay. No.
So now we can hear you.
Now the thymus gets shrunk.
Yes.
And so ideally we should have tried this vaccination on the younger population, because older ones are not going to develop the T-cell immunity that well, I suppose. I may be wrong. So we started at the wrong end, do you feel, or we should have started on the other end first?
So that's a paradox, right? So like Eddie was saying, that you want to boost the responses. So in that, I agree with Eddie that it's the older population that needs the help, right? So you want to boost the responses there. The younger population has their own ammunition, and they are very quick because the T cells will get educated pretty quickly. But you need, and they will recover. And as we see, 99% of the children recover, and 99%, a huge subset of them actually have much milder symptoms. Now, there are some children that are dying, but most of the adults are the ones who have more severe symptoms, and therefore giving them a boost and helping them is more practical. So with the children, the analogy that I give is like, if you were to do, just because you want your child to be an A student, you can't do their homework for them all the time, because they may look like an A student, but they're not really, you're not helping them. So the children have a very robust immune system. They have an ability to bounce back. So you need to let them, you have to pick your battles. So you do want to vaccinate them against really deadly diseases such as, chickenpox, smallpox, polio. But in seasonal diseases such as flu, unless they have serious respiratory conditions or other underlying health issues, children, you can let them take the bullet and they will recover. But it's not the case with perhaps the adult, where the thymus doesn't work as well. So you want to give them a little bit of a boost and help them.
But what I have found in this case in India this time is that number of young students, I knew my own students, quite a few died. A lot of children died. And if the theory is that they can withstand the infection, I don't think, I mean, I don't have the statistic, but if I am to come across so many students whom I knew, some of my BTech students died, some of my research students died, and I was a bit, and if this is the population I know, overall, if I extend the graph, the number would have been huge. And in India, it has been underreported. So I do not know whether, that argument that children can withstand better than this thing, I do not know. Because this is a very unusual case. It doesn't fit in with others, is my feeling. I may be wrong.
So I again don't know about India, but I do know that here kids in middle school and high school vape a lot, and vaping, which is basically was touted as a safer alternative than smoking. Tobacco has a lot of cross interaction with this COVID infection, and a lot of people who were smokers, they suffered much more than people non-smokers. Now, and that's why initially here we saw the kids that a lot of kids vape and they will never tell or admit that they vape. So I do not know whether that's the case in India. How many of them are vaping or taking other kind of so again, we do not know, and to just say that because those children died, it would be good to get some information on what else was going on with them, rather than making the assumption that and similarly, we've had, for example, perfectly healthy athletes who would collapse like on a basketball court because they had arrhythmia or something, which never was detected despite them going through all the health tests and things like that. So that's a difficult one.
Yep. Dr. Kannan, I'll just add one little thing, is because I've been actually monitoring these clinical trials quite a bit, So in any clinical trial, you stratify the groups, you take the age group. So there's under 50 and over 50, and then you start seeing the underlying infections or issues that they have, and then you start stratifying the data to see if there's some particular trend that is coming out, And what people were seeing, that people less than 50 handle this infection better compared to greater than 50, which is expected because of the underlying issues that people typically have. So when the vaccines became available, there was a choice. There's a limited amount of vaccine. Which group should we target first? So they said, ideally, let's protect the elderly because they would not be able to handle the infections in a better way. Now, there's one clinical study that my team did actually. We were collaborating with Merck, and unfortunately, that vaccine did not do well intramuscularly. But within that group, what we saw that if you had pre-existing antibodies to the spike protein, after getting this vaccine, our neutralizing antibody to the spike protein skyrocketed 1,000-fold. And what that piece of data told us was the vaccine that we were designing, which was based on a vesicular stomatitis virus as a platform, that it was a good boosting agent, but not a good priming agent. So these are the pieces of information that are going to come over a period of time. And there's this concept that I touched upon is the mix and match. So what may happen is we may go with the initial vaccine to generate a primary response, but then other modalities like the viral vectors, inactivated viruses may come back as a secondary response boosting. So there are some studies going on. I would say in the next 6 months, you will see that data emerging.
Yeah. No, it's a valid
And
point. I mean, you have an
yeah,
argument, right? I mean, I appreciate that. I mean, so that others also appreciate these other thoughts going on. Yeah.
yeah,
Raghuram has a, yeah, tell me. Yeah.
yeah.
Carry
And
on. Eddie.
one last point, I think Raghu asked a question about T cells. Yes, initially the focus was looking at neutralizing antibodies to spike, but there's data coming out on T cell responses, and we are seeing T cell responses to the spike protein as well as other proteins. In fact, the T cell response to the nucleoprotein will probably turn out to be more relevant because that's a very conservative epitope. So I would say just keep our fingers crossed. There's going to be so much data in the next 6 months that will educate us in terms of what are the components required for a good efficacious vaccine.
So I'll add to that yes, in a natural infection, you will see much more robust T cell responses. But there's one report that I've seen with the vaccine against the N protein, and in animals, apparently they made it very, very sick. So in vivo, when you get an infection, I think the way the body handles it, we don't understand completely. And when we try and make vaccines, of course, we are doing our best, but you may not. So I think that was the challenge with having a vaccine against the nucleocapsid protein. At least with the animal study, it made them really, really sick.
Yeah, and Aditi, what we have seen is actually is although people are using the non-human primate as a model, in our hands that is not a very predictable model. Actually, the hamsters is a much predictable model. We are able to demonstrate protection in hamsters, and that translates into a good response that we see in human. So again, we are still determining which is the best model. So lots of exciting data will come through.
Right, and that's all I'm saying is that science cannot be rushed, and
Yeah.
we are doing our best. And my main message really is for for the scientific community. Like I said, we are used to data being published and then it being updated and sort of revised, so we take it with a pinch of salt. But that's not the case for the general public. And so when you say something and then you go back and sort of try and change your message 3 months later, that's not very palatable to the general public. And for them, this is the first time they're consuming things in real time. And I think that's been the biggest problem.
Raghuram sir has a few questions and comments. Raghuram sir, could you unmute yourself?
Thank you. I have put my questions and comments mostly on the chat box, but I just wanted to say for others who might think that Eddie and Aditi are having a conversation from opposite perspectives. Both perspectives are needed for a effective public health system. It is absolutely natural for people who are developing diagnostic tools, developing vaccines, developing testing models, animal models, to want to bring their work into public utility as quickly as possible. It's a perfectly understandable sentiment. Uh, so people should come out and do things that matter and things offer them, and they should hope that they would come into public utility as quickly as possible. But that's not the same thing as saying that everything that is on offer has to be accepted. That's the perspective that Aditi brings. Okay. Uh, the skipping of phase 3 trials, the emergency approvals, the lack of rigor that is normally required in these things, and the ignoring of all the previous evidence about how many phase 2 passed cases have eventually failed in phase 3, ignoring of evidence about whether too much focus on humoral immunity really gives us the kind of public health protection that we need. Are we only focusing on individual protection? What happens to transmission? Is every vaccinated person becoming a super spreader because he may or may not know that his vaccination is not designed for stopping transmission? So these are all issues that are extremely important uh, and have hit the public in a manner that they have never been prepared for something like this. And that's the point that Aditi is making. And uh, without necessarily thinking that they are actually, arguing from totally opposite ends of the spectrum, the public needs to understand that both these perspectives actually coexist in the normal scheme of things. If you don't allow them to coexist, if you prioritize one over the other, you are compromising. And that's really the problem. And the other point that I just hinted, but I wanted to make, is that this whole thing about selective immunization versus universal immunization has been almost forgotten in the manner in which scientific evidence is presented to the governments. Everybody thinks every available vaccine has to be given to everybody who's born on Earth. This is completely nonsensical, scientifically speaking, even in terms of public health. There are very well established rules of public health which are being deliberately underplayed both by the industry and by the governments. In Indian vaccine policy, which was actually adopted a few years ago, a decade ago, under pressure from Supreme Court case, if you do a keyword search, you don't find selective immunization anywhere in the entire document. Any vaccine is only universal, nothing, no discussion. So there are a lot of politics of language, politics of presenting science to the public in a manner that science is not prepared to understand, public is not prepared to consume, the manner in which you and me as scientists would normally consume. There are a lot of caveats attached to everything that we say in science, whereas the general public may not always look for the caveats because they are desperate. They are looking for magic solutions. So we need to have a healthy balance of both these perspectives rather than you versus me and this versus that. That's all I would like to say. Thank you very much.
Yes, you have
Yeah, thank you,
summarized it.
Rafa. Very
Sorry.
well. Go ahead, Professor Aditi. Please go ahead.
No, I think Raghu has really summarized that well, that we are not really the kind of, I don't want to repeat. Basically, yes, I agree. And I also agree that we have to have a healthy discussion. We basically, there is just no discussion, at least here, at least in the US, I feel now with the vaccine mandate, even though the federal government says no mandates, every local government, every business, my university has a vaccine mandate. And I just feel that that's incorrect. And they are demonizing anybody who says that they don't want vaccine. They're being socially ostracized. They are, there's just so much of pressure. Like I just said, I've had heard of cases where they are forcibly taking away their parents, or they're saying they're going to take away the kids because you are not vaccinated. I mean, that's just going to extremes, and it's a dictatorship.
I think, Nitika, let us try to wind it up now. So,
Sure.
quite.
See, if permitted, I have just one small question.
Okay.
Both for Eddie,
Yeah.
and Aditi. Yeah. So actually, Professor Raghuram is focusing on that universal vaccination. Yeah, I was just thinking whether with so many different kinds of vaccines, including mRNA vaccines, if we are giving an opportunity for the virus to really mutate more under the vaccination regime, whether so many diversified vaccines may actually provide the opportunity for the virus to become stronger. Is there any likelihood of that? And whether we should only use only one kind of vaccine at a time, in the way for antibiotic resistance, they follow a antibiotic regimen.
That's a great question. I think this is a possibility to consider. That would you drive more variants to appear because of selective vaccination against spike protein, right? So now there is a selection pressure on the virus, and it's a virus that mutates pretty often. So would it mutate and you will see more variants? The other thing to just keep in mind is that there are a lot of diseases that, such as that I study, for example, the inflammatory bowel disease, where the treatment, for example, is to give anti-TNF alpha antibodies because these patients have a lot of TNF alpha. Now, over time, within few months of giving anti-TNF alpha therapy, 40% of the patients become refractile. What that means is that they develop antibodies against anti-TNF alpha. So if you have a constantly high amount of antibodies, say against spike protein in you, you do not know whether your body will make antibodies against spike protein, and you may become refractile to that treatment. So that's something to keep in mind about, boosters and things like that. We are not even, we're pushing for things without fully understanding the implications.
Karan sir, can you please unmute Lakshmi?
Aditi, I think you have to unmute her because you gave her the link. Aditi, you try unmuting your mom. Yeah.
I can't seem to unmute
because
her.
I'm also getting a signal I can't do.
Same here.
Oh, so she unmuted herself.
Yeah, please, yeah. Let her go ahead, yeah.
So do you have a question?
Aditi, this is Ritu. Mummy is now it's time for her eye medicine, and then she's going to bed.
Oh, okay. Okay, I'll answer her question on another call.
Okay. No,
Thanks.
you can answer it. She can hear you. She can
But
hear
she
you.
didn't pose the question, so I can't answer it.
Oh. Do you have a question to ask? Okay, she'll ask you later. Huh?
Okay, no problem. Thanks.
Aditi, she must be giving you her blessings, for such a wonderful talk.
Well, it's late and she's diabetic, so she's got her.
She must be very, very proud of you, actually, of talking such an important topic, which is baffling all of us. I'm sure she's very proud of you. Utpal, I just wanted to respond to your one point. Sorry, is your mom saying something?
No, continue.
Yeah. Utpal, this is a difficult. Your question is a very difficult one. I'll tell you why. You see how does a virus evolve variants? Because it's under pressure, and it's under pressure because of its survival, because of the immune pressure. The question comes is, do you vaccinate and exert another immune response level and exert more pressure on the virus, and the virus actually then variants where it starts producing more mutations? And it's so difficult to answer that question, because the other part is, don't vaccinate the person and let the virus take its own course. I'm not sure how we would be ever able to answer that question. But we definitely know that an immune response to a natural infection is far superior. So, we have to let time and science tell us what choices to make. And of course, it will be the individual who will have to make a choice.
Thanks, Eddie. No, I am not against vaccination. What I was thinking about the regimentation in the way antibiotics are regimented. In India, there is no any regimentation. Doctors prescribe whatever antibiotic comes to their mind or some kind of commercial gain they get. I was thinking whether we should go one vaccine at a time, so that we don't give enough opportunity for the virus to mutate more. But of course, I understand it's really difficult, and there are other questions of supply versus demand, and many different kind of manufacturers stepping in to fill up that gap. We really have big challenges. Yeah. Thank you. Thank you.
So thank you, Professor Aditi. It was a very nice talk, and I thank all of you for a very good discussion board that we've had today. Because it's very important as members of scientific fraternity to keep discussing the science and to also question it. Because only when we question does science also remain ethical. So I think that's very, very important. I thank you all for putting across very nice comments and questions to Professor Aditi. I think Raghuram sir still has a few more questions to go. And if
we can take the discussion offline, and happy to.
But I must thank. I think it's. I must thank Nitika for managing so patiently.
Not at all, sir. My pleasure. My pleasure, and it was wonderful, sir. Thank you. Thank you.
Kanan sir, I have a small proposal.
Tell me.
Yeah, I think if you allow all people to unmute themselves and give a round of applause for this great speech, it will be fantastic.
Yes, I agree. Thank you so much, Aditi.
Thank you very much. And I also thank Mohit for joining, my cousin from, I don't know where he is actually, in Mexico or in Canada. I have no idea.
Thanks, Aditi. I saw somebody from the UK also joined, your relative.
Oh, maybe
Kamalini joined, I think.
Yes, my aunt, my mother's younger sister.
Yeah. So I saw her. So
and you, of
anyway,
course,
it was a lovely.
Mohit, right?
Yes, I do. Lovely, and it was nice to meet all our old people. Normally, would not have met. I think it is so nice seeing all the 80s crowd with us. Am I right, Eddie?
Yeah.
Yes.
And what I like. Carry on. Yeah.
I just saw Vivek and some other people join. I don't have a full screen, so I can't see who all joined, but I do want to thank everyone who joined and for staying on for a long time.
Vivek is also there, Vivek Verma. You remember him?
Hi, Mohit.
Both the Viveks were there, sir.
Okay. Lovely. And I must thank my batchmates also. Many of them joined. Vishweshwar is there. Rohit is there. Thank you so much. Okay.
Thank you, everyone. And thank you.
Thank you, Kanan sir, for putting this together. Thank you, Kanan sir.
Yeah. And please join for the 23rd one. It is going to be fun. Blood grouping and how various groups. I mean, he's really good. He works by hand. So you will really enjoy.
Definitely.
Thank you, and have a good day in America and a good night in India.
Yes, good night, everyone.
Good night. Thank you. It was wonderful.
Thank you, everyone.
Bye. Thank you, Nitika.
Okay, sir. Thank you so much. It was a very, very nice session. Very interesting. And I think the discussion was best part.
Yeah. I think it served the purpose. And all were serious people. I'm not worried about the crowd. I was more interested in people who really enjoyed. So it was really nice. Okay.
That's true.
Take care now.
Okay,
enjoy
sir.
with your children.
Bye.
Yeah. Bye-bye. Bye-bye.